A very good afternoon to all of you. Myself, Dr. Vikas Sharma, and I'm working as Associate Professor in Department of Molecular Biology and Genetic Engineering, Lovely Professional University, Fagwara, Jalandhar, Punjab, India. So the topic of my research is plant tissue culture based plant specific in vitro approaches to screen beneficial PGPRs for enhanced caulogenesis and rhizogenesis in endangered medicinal plants. So in this present research, I'm going to discuss about some of the problems which are associated with the agricultural component as well as the in vitro micropropagation. First of all, I will talk about the medicinal plants. Medicinal plants are one of the most important bioresource right now as far as the health components and the nutritional components are concerned. In the COVID era also, we have seen the applicability of medicinal plants in the immunity boosters, in the sanitizer development also. So they are very important, and we know that there are so many important medicinal plants which are at the verge of extinction. And micropropagation or plant tissue culture are one of the most important methods with which we can go for the improvement of the plants as well as their introduction into their natural habitat, like in the conservation of those medicinal plants. Now, but if we talk about the micropropagation protocol development of the medicinal plants, there are so many problems. One of the problem is also associated with the phytoconstituents or bioactive content of the medicinal plant, which interfere with the media components and make it difficult. And sometimes it has also been seen that um, it also the process of organogenesis is also very much difficult to establish in vitro conditions. We know that the microorganisms, they are not required at all in the plant tissue culture methods, and if they are present over there, they will spoil the culture and they interfere with the success of the micropropagation protocols. But at the same point of time, we know that plant growth promoting rhizobacteria are the microorganisms which contribute towards the growth of the plants. And there are so many ways, mechanisms identified with which the microorganisms they contribute in the plant growth, like the nutrient acquisition, nitrogen fixation, phosphate solubilization, iron sequestration, biocontrol. So many have been identified. So present research basically involves the development of a method of an in vitro assay through which we can screen such type of beneficial plant microbe interactions. Now, this is very much important because if we know that a particular microorganism is suitable for the growth of a particular plant, so it can help in the establishment of the in vitro raised plantlets, because we know that the last stage of the plant tissue culture methodology, the hardening, uh, acclimatization and field transfer, that also decides the success of the plant tissue culture protocols, and we have seen so many mortalities in that case. So these plant microbe interactions, they can help us in the screening of the microorganisms which can contribute towards the enhanced caulogenesis, which is the shoot multiplication, and the rhizogenesis, which is the rooting. Because if the plants in vitro raised plantlets, they are having the high root vigor, the percentage of hardening and acclimatization that can be enhanced, which can help in the establishment of the field transfer of the micropropagated raised plants. So obviously it is going to reduce the mortality. And apart from that, this can also be extended for the development of plant specific bioinoculants for the fertilizing activities in the vegetatively propagated plants also. So that is basically the core component of this present research, to establish an in vitro assay system using the plant tissue culture methodologies, and it is of very much importance, and we are working on it. We know that there are so many shortcomings in our methods, but the suggestions from the scientific community definitely help us to modify it to establish it into a better way. Thank you so much.